Thank you. So yes, I am just a measly second-year PhD student, so I'm going to be like presenting this lovely idea of what the next few years of my life will be, and hopefully it will pan out like that. Fingers crossed. So also, which was really good and wasn't planned in any way, shape, or form, mine is very like statsy and you know, stats applied to biology, just like Amy's talk herself. So perfectly timed. I am also at the University of Edinburgh as well, but I am in Edward Wallace's lab, which is more transcriptomics, fundamental cell biology, and I'm part of the East Bio PhD program. And this is a really nice little photo of the lab with Edward Wallace at the Wallace Memorial, as in William Wallace, up in, or just round about Stirling, a little bit just out Stirling. So it's quite cute that we all went as a lab. Anyway, so what am I going to be talking about? First, I'm going to give a little bit of an introduction of who I am, which is going to be a very short introduction because I'm not that much right now. And then I'm going to talk about Bayesianness a tiny little bit because, you know, sometimes it can be a little bit confusing if you're in the stats world and you've picked a side between frequentist and Bayesian, then fair play. But I will just elaborate my little hint of knowledge, and then talk about the biology that I'm applying my Bayesian stats to. Right. So yes, a little bit about me. I originally wasn't a statistician and wasn't definitely not a biologist. I was a physicist. My undergrad and my master's are in physics. One of the first things that I did outside of actually my degree was do some really cool, oh no, it's not going to work for me. That's really sad. Anyway, that was meant to be an animation of what happens when you start to fall into a black hole. So as you get closer and closer to like the Schwarzschild radius of the black hole, where it's like the point of no return, this little bubble you see is the whole universe. You see the way that light bends around the black hole, you actually see the entire universe in sort of like a fish lens, which gets smaller and smaller and smaller as closer you get to the Schwarzschild black hole. But it was quite fun when I was looking back over my old notes and I was seeing this little animation, but apparently it's not going to play for us, which is a shame. Then, after doing some theoretical physics, I moved on to more observational. So this is what I did for my master's. It's picture, I don't know, can you see anything? No. Oh, can you see my mouse? I think you can see my mouse. This is a photograph in essence taken by a radio telescope over in Hawaii, which, you know, I was in control of at the time. And it is a picture of two baby stars in essence that are in the very, very early stages of forming. One's a little bit older than the other one. So this one, the brighter one on the left-hand side, is a little bit more active, and it's got a lovely little outflow, which is what this red lines are denoting, that it's like getting quite energetic, starting to actually burn some hydrogen and fire out some outflows. And then right beside it, you can't see it quite as well, it's a little bit more of a blurry blob to the bottom right of it. And that is a younger star, which hasn't even started properly hydrogen fusing. It's just like collating mass from the surrounding cloud. And then maybe one day it will properly explode, and according to my measurements, it should be really massive, massive star, which billions of years in the future will explode into a black hole. And that's the first time anybody actually viewed this little star. And so I named it, but it was actually a really stupid name because it had to be named by the nomenclature of the telescope, which is SDC-18b. This is SDC-18a, which was seen before, and SDC-18b. It's my little contribution. But after that, I thought, I'm not going to do any more physics. Well, I'm not going to do any more astrophysics, and move to biology. What? Madness. And I'm now in the Edward Wallace Lab, who Edward paid an artist to make a lovely little depiction of what the lab does. In the background, you've got yeast cells just sort of casually growing and multiplying, and then you've got like some sort of sequencing machine and lots of graphs and scientists looking very important, doing cool things. And apparently I'm now one of those scientists, and we are looking particularly into how yeast cells react to different stresses. Sometimes that's just for our own interest in terms of fundamental cell biology, and sometimes it has like a medical inclination. For example, Cryptococcus neoformans, which is quite a serious, or can be quite a serious fungal infection for those who have some sort of immunocompromisation. It has, like, Cryptococcus neoformans has this weird ability that as soon as it gets into your lungs, it can transform into what are called Titan cells. So it just blows up. And that means none of your like macrophages or anything, they can't engulf it. And so it stops, like the infection can stay for a long period of time in your lungs and can't be destroyed. And then slowly it starts to bud off into smaller little fungal cells that then can get into your bloodstream and start infecting your body, get into your nervous system, get into your brain and cause really, really serious infections. And so a lot of our research is trying to understand how, well, some of our research is understanding how or what triggers or what changes in these yeast cells to make them blow up into these Titan cells. And specifically when we're talking about what changes happen in the cell, we're specifically interested in like the transcriptome, what happens, what RNA transcripts does the cell make, what transcripts does it try and store away, what transcripts does it try and destroy in response to its environment. Right, so we've got a biology background, and now I'm going to do a little bit of a stats background. So here's Reverend Bayes again, which you saw at the very beginning. I don't know how familiar people are with stats, but his most famous work, Bayes' theorem, which is the idea of conditional probability, separating or swapping, in essence, swapping the conditions around. So if you've got A conditioned on B, you can make it B conditioned on A, given the probability of A and B. I've forgotten which way I've done it now. And in and of itself, that doesn't seem to, mean a huge amount. But when you're applying it to data analysis, it can be a really, really powerful tool. Because if you're trying to work out, for example, in the equation, the second equation, if you're trying to work out mu, like the mean number of, for example, transcript counts in a cell, you're trying to find out what the mean number is, but you've only got limited data. So all you can say, in essence, is what is the mean, or the global mean, given the data that you have. And that's kind of a difficult thing in itself to calculate. But if you swap it round and you say, well, if I have a whole different range of parameters of mu, what's the probability of getting the data that I have? So you swap the question around, and suddenly it's a little bit more easier to answer. And then with that, once you've swapped the question around and started to have something you can actually tackle, you can start creating answers to the questions that you actually want, which is, what's the expectation of the mu? What's the expectation of the mean number of transcripts in these cells, given the data that I've collected? Unfortunately, there's some controversy around how you actually do Bayesian analysis, because with each likelihood, you have to have these prior distributions. You have to come with some idea of what values these parameters, this mu, this average, should have already, and then add the data that you get and combine them together to give the posterior distribution. And the decisions around the actual prior distributions can be sometimes controversial. But anyway, that's the general thought and logic around Bayesian analysis. So let's go back to the biology, and then I can fit in why and how I'm using Bayesian analysis. So back to what these yeast cells are doing. They're in different environments, sometimes depending on what stresses we introduce them or stresses they might introduce when they go into a new environment. There's a whole plethora of different stresses there given as an example, and we're interested in what happens inside the cell once it's exposed to these different stresses. Are the responses similar? Are the responses completely different? And again, typically the responses of these cells are on the form of, well, ultimately proteins, because proteins are typically what proteins it makes and what proteins it has available typically dominate its response to its environment. But ultimately, those proteins, the volume of protein that's in a cell is dependent on what creates these proteins, which is RNA, or using what's encoded in RNA. So whenever these cells react to their environment, normally one of the first things that change is the RNA levels, because the RNA levels then affect what ultimately the protein production levels and the amount of protein that's in the cell. So how do we start investigating these RNA levels? Well, for a lot of you, you might already know, but if you don't know, I'm going to do a quick overview of how you start counting the transcripts, the volumes of RNA in a cell. I mean, the first thing you do is you mush the cell up, or the whole sample of all your cells, you mush it up and you destroy the cell and try and extract just the RNA on itself. Then, unfortunately, most high throughput sequencing machines can't deal with like whole transcripts, the whole length of transcripts, because they can be pretty long. And so you have to first chop it up. And then once you've chopped it up into smaller fragments that the sequencer machine can actually handle, then you need to amplify it a bit more, because sometimes, you know, especially for low abundant transcripts, if you only have one or two in the cell, even the best sequencer isn't really going to be very effective at detecting them. So you have to amplify what you already have, like 10-fold, 20-fold, and then count that and hope that you can inverse the amplification to try and guess what the actual original samples were. And then you've got to do all these other fancy additions to the transcripts when it's talking about like the adapter ligation. These are just extra bits that the sequencer needs to have in order to be able to manipulate and detect these transcripts. So all you need to care about is, you know, it's not as simple as taking a photograph and counting these things. There's a lot of steps that happen between whatever the yeast cell's doing and when you actually get something out of your sequencing machine. So with this, with all these different inputs and different stages of counting these transcripts, how do you actually deal with all these sources of errors? You've got things about the actual fundamental biology that's going on that might complicate things. Maybe, like I said, maybe some of these cells have actually become Titan cells, and so you suddenly get a lot more transcripts per cell. And so not necessarily, there isn't really like any particular genes that are overly expressed and has lots of copies of. It's just that the whole transcriptome has just been blown up. And you've got to try and separate that from like the noise and the errors that happen in all the different steps of the sequencing machine itself. So how do we start trying to account for these different sources of noise? Well, scary symbols time. You can, in a Bayesian framework where I was talking about when you're trying to find out the outputs of a specific parameter, I said mu last time. When you're using Bayes' theorem, you have you're separating your conditions into, in essence, like different multiplications, and each multiplication you can think of like a separate model of a certain section of the problem. So in this more complicated model, you can actually see that each square box that incorporates different parameters is actually dealing with a completely separate section of your problem. For example, if you're thinking about the cells themselves, maybe each cell might be a different size, or just might not be, I don't know, might not be happy for some particular reason. You need to have certain parameters that try and model just errors with respect to that cell. Maybe for particular genes might be naturally highly expressed. Maybe it's something to do with, you know, the actual energy production or something like that, proteins that are associated with energy production, or proteins that are associated with building proteins. They're always like quite highly expressed. And so specific genes, you might want specific parameters that naturally give high accounts for those particular genes. And then if you're trying to account for something to do with a particular sequence of like an actual batch of sequencing, say that was a particularly poor batch and there was quite a lot of errors in the amplification stage or whatever, maybe you want parameters that try and count, or that try and take out some of this error so you can compare multiple batches, multiple sequences. And I'm not going to talk you through this particular model, but that's exactly what this model is doing. It's trying to think of each part separately and then multiplying it all together using Bayes' theorem, so that you have a much more easily tackled solution. So let's say you've done all of that, which somebody has. I've done a little bit, but not enough to make me feel brave enough to show it to everybody. But what do you get out of it? Well, let's say you've removed a lot of the variability because of batches, because of biological conditions. Then what's the remaining variability? Let's see what genes have particular high variable transcript numbers, because maybe that high variability is actually biologically significant, and the reason why this transcript is suddenly, you know, sometimes highly expressed, sometimes lowly expressed, it's because it has a specific use in a specific condition. And doing models such as what Catalina Vallejos has done, you can start highlighting, putting down a threshold saying, oh, I want to look at all of the genes that seem to be highly expressed, or I want to, well, that seem to be highly variable, and then possibly just specifically looking about particular genes that are expressed highly in certain conditions, or even lowly expressed in certain conditions. Yeah, and so what has that got to do with me? Well, I'm trying to do a similar certain model, but for even more, arguably more complex situations where I don't actually have a slide for this, and this is silly, but on top of the general variation that you have to when you're, for example, looking into protein or RNA protein interactions, and you want to know, is this RNA transcript often bound by this particular protein? Or then you've got to start to account for like other forms of variation. So my project is to try and use, in essence, Catalina Vallejos's work and add, again, another almost independent module on top of what was previously seen there. But there's been a plot twist, as there so often is, and it's something that's affected all of our lives, with the obvious COVID-19 outbreak. And I've currently put, well, literally yesterday, put aside all of my research into fundamental cell biology, and I'm trying to help collate and analyze some of the outbreak data, specifically in Africa. As you can see, I put this particular map up not to scare anybody, but to try and highlight the fact that if you look around Africa, it doesn't have, it's either at very early stages of the outbreak / doesn't necessarily have a huge amount of coverage right now. And so I'm trying to help with the University of Edinburgh's TIBA project in trying to help inform or help African countries develop policies to deal with the inevitable outbreaks in their own countries by accurately modeling their outbreaks. Right. That is me done. Does anybody, obviously I need to thank my supervisor and my second supervisor, Guido Sanguinetti, and all the people that are associated with Edward Wallace's lab. Feel free to contact me. I love talking about stats. So if you've got stats questions or you just want to argue with me about why frequentist is so much better, then yeah, I'm happy to tweet me, email me, whatever. Any questions?